thanks for the organizing committee for inviting me for this presentation. My topic is preparation and in vitro in vivo evaluation of metformin hydrochloride rectal dosage forms. So, so the outlines I'm going to cover in this presentation. I will give a brief introduction about this work. And this introduction includes rectal drug administration, type 2 diabetes, anti-diabetic drugs, particularly the metformin hydrochloride, also rectal suppositories, and actually Tina enjoyed us yesterday about this topic, so she did a lot. And I will talk about the experimental work we did in this research project, which include preparation of different metformin hydrochloride rectal suppositories and the characterization we have done and the bioavailability testing. And we'll mention some of the results we got and we will end by conclusion. So rectal drug administration is a good alternative route for oral and invasive drug delivery. The main advantage of this drug delivery system is it is non-invasive, easy to use, and can be applied in several situations, such as when administration by the oral route is intolerable in case of nausea, vomiting, gastric pain. Also, when patients are uncooperative or unconscious, when access to the parenteral route is difficult, like in cases of in remote areas or in safety. Also in ambulatory patients when repeated intramuscular or intravenous administration is needed. In addition to that, the rectal drug administration avoids at least partially the first pass metabolism because, as you know, the vascularity of the rectum the blood can goes directly to the general circulation or can goes to the hepatic circulation. So the drug can partially be saved from the first pass effect. Also improves the stability of the drug undergoing gastrointestinal degradation, allows the administration of unpleasantly tasting or smelling drug, and this would be good and nice for children. Also allows the lymphatic circulation to assist in the absorption and hence improve the bioavailability of administered drug because, as you know, eventually the lymphatic circulation poor in the blood circulation and the drug absorbed through lymphatic system will be shown later in the general circulation. Type 2 diabetes, it is the most common and highest prevalence type of diabetes mellitus. Arab countries are reporting high rates of this disease. According to International Diabetes Federation, Middle East and North Africa, 415 million people have diabetes in the world. Among them, more than 35 plus million people in the MENA region, and this number is going to be doubled by 2040. Kuwait, the country in which I'm living and working, is ranked third on a global level and first in the MENA region with high diabetes prevalence. Approximately 1/4 of the population are suffering from diabetes. Patients who are admitted with this type of disease in Kuwait are younger compared to the same category around the world, and the justification for that may be the increase in obesity and the change in the lifestyle. One of the drugs used to treat type 2 diabetes is metformin hydrochloride, and I will talk a little bit about this drug because it is a candidate in my research. Metformin hydrochloride features as a current first-line pharmacological treatment in almost all guidelines and recommendations worldwide. It improves glycemic control by reducing hepatic glucose production, increase insulin sensitivity, and reduce intestinal glucose absorption without increasing insulin secretion. It is particularly useful in overweight and obese patients, so it is good for us to be considered. I mean, the drug is considered in my research for this reason. Also, it is effective both as a monotherapy and in combination with other anti-diabetic drugs and insulin to improve glycemia. One or some of the advantage of this drug, it doesn't pose the risk of hypoglycemia. It doesn't cause hypoglycemia. It doesn't cause weight gain. It improves the lipid profile and counteracting the cardiovascular complications associated with diabetes. And very recently, a paper came in 2016 saying that this drug has shown benefits in reducing cancer risk and improving cancer prognosis. So it is a multiple use drug. Also, it has shown to be safe and effective for treatment of this of pediatric patients suffering from diabetes. But on the other hand, there are some limitations and disadvantages. This drug, if taken orally, it is slowly and then completely absorbed from the GIT. The absolute bioavailability is about 50%. Oral bioadministration is accompanied by GIT adverse effects. Also, the large tablet size of this drug and the lack of tolerable liquid formulation in the market make children and certain patient categories with type 2 diabetes in dire need to alternative dosage form of this drug. Because if the dose is high, like 500 milligram, maybe increased to 750 milligram, and the people working in industry to prepare such a tablet, you need to add excipients like disintegrants, lubricants, binders, fillers, and the tablet weight can go up to 700 maybe milligram to give this drug for kids. It is difficult actually. Also for those people who are suffering or who cannot swallow these large tablets. So this alternative could be the rectal drug delivery or rectal suppository. So now I'm confident that this drug, we should search for an alternative delivery system for this drug. And I quite confident that rectal suppository could be this alternative. Considering that this kind of dosage form is acceptable in Arab countries, where most of the dosage forms or most of the drugs are formulated as oral side by side with rectal formulation. So we do not have problem with the rectal administration of drugs. When we talk a little bit about suppository, it can be very simple, straightforward formulation, or it can be a complex formulation. Many factors affecting the drug release, like formulation, physicochemical factors, physiological factors. All of them influence the absorption, distribution, and pharmacokinetic of rectal administered drug. But fortunately, from my experience, by proper formulation, optimization, and selection of the appropriate pace, also selection of the release and penetration promoters, some of these problems could be solved. And recently, reports state that the absorption of sodium ampicillin in the presence of 2 to 3% of sodium caprate, which is absorption promoter, is comparable to intravenous administration of this drug. So the bioavailability can go high and be comparable to the injection. So the overall objective of my study is to develop rectal dosage forms of metformin hydrochloride using suppository bases like Witepsol, Sapusir, Mesa. We use different grades. Hydrophilic bases like polyethylene glycol, different grades and different ratios. And from my perspective, this dosage form is assumed to avoid the GIT intolerance, improve the patient compliance, and enhance the bioavailability, and can be used as a good alternative for pediatrics, geriatrics, and other category of patients. After that, I am going to do physical characterization for the formulations, like mechanical strength, melting time, penetration time, content uniformity, and in vitro drug release. And based on the results, the formulations showing the best results, satisfactory results regarding the physicochemical characterization and the drug release, we choose them for bioavailability testing in human volunteers. So this one of the good things is this in this study, we used the human volunteers and using validated selective precise analytical technique applying LLC, MS. So the experimental work, first of all, we did selection of the suitable base. As I mentioned before, the base we selected were fatty bases as well as hydrophilic bases. For this purpose, 9 semi-synthetic fatty bases, including eutepsol, sapocere, and masa, and 2 formulations containing polyethylene glycol bases we used. Cacao butter base as an oily base, we excluded it because of the polymorphism, and also the glycerogelatin base was excluded because of the rectal irritation, because we are going to give them to human volunteers. This table summarizes the composition of the 11th formulation we used. After that, we have to determine the displacement value of the metformin hydrochloride, because as you know, when we prepare rectal suppository, we calibrate the moles by volume. But when we're gonna calculate the amount of the base needed, we calculate it by weight. So we have to calculate the displacement value. This value is defined as amount of drug by weight that displaces 1 part by weight of a specific base being used for the manufacture. And searching in the literature, we came out with this equation by which we can calculate the displacement value for each base. After that, we started preparation of the suppository bases. Again, based on the results of this displacement value, we can calculate the amount of the base required to prepare certain numbers of suppositories. The fatty bases where I'm not going to go in detail about the preparation of the method of the preparations, just to save time and to make it short. After that, we did characterization for the prepared formulations. The characterization included visual observation or visual inspection for color change, clarity, fissuring, pitting, oxidation, sedimentations, all these things, and technical evaluation for content uniformity, weight uniformity, mechanical strength, penetration time, disintegration or melting time, disintegration for hydrophilic bases, and the melting time for fatty bases, dissolution testing in phosphate buffer using a spectrophotometer technique. And after that, the analysis and the bioavailability study of the chosen formulation. As I mentioned, my work in the lab stopped at preparation and the characterization of the suppository of the different formulations, and after that, we choose 3 formulations showing best characteristics and the highest amount of drug release, and we send them to a company. And this company make the registered company. They are specialized in bioequivalence and bioavailability determinations, and they have professional people. So we just sit together and put the protocol for this study, and we just paid money, and they take care about everything. The pharmacokinetic study, the developed method applied to pharmacokinetic study following a single oral or rectal dose of metformin hydrochloride. Actually, about 12 healthy adult volunteers were participated in this formulation. We have 4 formulations, 3 rectal formulations compared with the oral tablet containing 500 milligram of the drug, and we used it as a reference. And the study was performed according to the EMA, ICH, GCP, and FDA guidelines. So there is no problem. They have the ethical committee, they have everything, and they are professional, and they do a good job. These are the results of, not the result and the condition of the experiments. And these are the results we got from physical characterization. This is the content uniformity, and we can see that there are some variations and in the content uniformity. The amount of the drug ranged between 92 to 105, which is acceptable. This is the penetration time, and this determine how soft the base is, because for fatty base, they gonna melt in the rectum and before releasing the drug. And this is for melting and disintegration time. And we can see here that the hydrophilic bases, formation number 10 and formation number 11, show the highest disintegration time, because maybe the hardness or the higher melting point, and they have to dissolve in the rectal fluids, unlike the fatty bases that are melt at the body temperature. The formulation chosen were formulation number 1, 2, 3. The first one containing Sapusir H12, the second one containing Sapusir AP, the first one containing Witipsol, the second one containing Sapusir, and the third one containing Masa Estranium, and all these were tested against the reference tablet, which is a pseudophage tablet, 500 milligram. And actually the results make sense, because since the drug is a hydrophilic in nature, so the release of the drug, according to the theory of opposite mentioned in the literature, the hydrophilic drug is released better from hydrophobic bases than from hydrophilic drug. Because as you know, the principle of like dissolve like, so the hydrophilic drug is retained in a hydrophilic base, but released better from a hydrophobic base. These are the pharmacokinetic parameters. This is for the reference, the oral tablet. And this for the formulations. And you can see max, you can find Cmax here for all formulations. And you can find out, or you can figure out that the Cmax in our formulation is maybe 7 to 8 times higher than that from the tablet. The Tmax is 3 hours in reference, but just 25 minutes, 15 minutes and 30 minutes and 25 minutes in our formulations. This is the AUC value, the area under the curve, from 0 to 24 hours. And this is the K elimination, and we use the AUC value to calculate the relative bioavailability. From the results, we can see that one of the formulations, the first one containing Witipsol, showed just only 76%, but other formulations, formulation number 2 and number 3, showed comparable results with the oral tablet. These are the results of the Cmax. This is the Tmax, and this is the drug plasma concentration at different time intervals. And these are the results of the relative bioavailability. In conclusion, I can say that metformin hydrochloride, the antidiabetic drug, incorporated in suppository bases were successfully prepared and characterized. The fatty bases in general showed better pharmacotechnical characteristics compared with the polyethylene glycol bases. Sapusir AP and Masa Estranium B containing formulations showed comparable bioavailability, higher Cmax and shorter Tmax compared with the commercial tablets. These two formulations could be a potential alternative for treatment of hyperglycemia, especially in case of emergency for pediatrics, geriatrics, and other category of patients when parenteral route and trained health care provider are not available. Those scaling up or scaling down can be tailored according to patient need and can be prepared extemporaneously, and this is a good thing. Keeping in mind that I did not add any absorption enhancers or release enhancers. I just use the base only, and the drug was dispersed in this formulation. But I am sure if I add any promoter enhancers, these results could be changed. However, I do not want to be optimistic that far. Considering the small number of human volunteers participated in the study, the obtained results should be considered as only indicator. The local intolerance, along with the advanced toxicological studies, the effect of added permeation enhancers to maximize bioavailability are still needed to develop the proposed formulation in a large scale. The acknowledgement for this work, this research administration at Kuwait University and the Kuwait Foundation for the Advancement of Science for their support to attend this meeting. Thank you for attendance, thank you for attention, and your questions are most welcome.
Now this house is open for discussion or some questions regarding this rectal suppository of metformin drugs. Any questions? I have a couple of questions.
Sure.
Okay. My first question, this normally for rectal delivery, the drug should be given for small amount. But metformin, it normally requires, see, 1,000 to 1,500 milligram drug. Maybe 500 to 750 milligram drug per tablet. So it's difficult to give in this suppository mold or suppository base. So how will solve this here?
Actually, I can say the opposite. One of the advantages of rectal drug administration is you can incorporate large amount of drug, because the suppository base for the suppository weight for adults, for example, is 2 grams. Or maybe more than that. In 2 grams suppository weight, you can incorporate up to 1,000 milligrams of the drug. So one of the advantages of the rectal suppository is to incorporate higher percentage of that. So if you have a problem with high doses, make them rectal suppository.
Okay. I'll go back to your this
Sure.
pharmacokinetic data.
Mm-hmm.
Bioavailability and pharmacokinetic data where Cmax and KEL elimination rate constants are given.
Just a second.
In your bioavailability study, it is shown that this AUC, area under the curve, is almost comparable with this normal tablet and rectal suppository.
Mm-hmm.
But it is shown this elimination rate constants, it is increasing. If elimination rate constant is increasing, the biological half-life will decrease. And if biological half-life decrease, then how this hypoglycemic effect of this metformin will continue? Because nowadays, this metformin is available in the market as sustained release. And it is required also for 24 hours, this sustained effect is required. So if,
Good.
before that,
Good.
just before, I think 2-3 slides before,
This one.
last, next,
This is an,
next, results,
should be this.
results, it comes under results.
Oh, sorry. Yeah. This one?
Yeah, here.
Yeah. Sorry.
Yeah. See this area under the curve, AUC, it is almost comparable, but this this KEL, this which is the elimination rate constant, it is increasing, gradually increasing. So if this area, I mean, elimination rate constant is increasing means half-life is decreasing. Half-life decreasing means bioavailability decreasing. Bioavailability decreasing means hypoglycemic effect also will decrease.
Actually, I do not believe in that, because for the absorption, we have extent and rate of absorption. So sometimes you may have high extent of
if absorption increase, okay. If Cmax is increasing, okay.
Yeah.
But this
This
it
is the
will
rate
go
for
to the
Cmax.
peak and again it will fall. But when it will fall, then this hypoglycemic effect will produce. See how you will maintain the hypoglycemic effect.
Yeah, that's why we are using this dosage form for emergency cases. In case of hyperglycemia, we can use it right away and it can produce its effect within 15 minutes.
Okay. Any more questions on this? Thank you very much.
Thank
It's a novel idea, of course, but yes, relatively it has to be again improved with this basis, because the selection of base and this the permission enhancer and penetration. There are a lot of works.
Yeah.
There. So I hope it will carry.
Yeah. Thank you.
Thank you very much.
Thank you.